We need to keep on time and we need to get going. My name is Stephen Deeks. Have been sort of helping Sharon and others put these forums together over the past several years, and they've all been quite successful, including today's. And then our goal today now is, after having discussed, all day we've been discussing sort of the translation of novel, clever ideas to get the virus under control, to identify targets, to develop immunotherapies. And we've now sort of entered an era, I think, of what I call experimental medicine, in which we're basically using these tools as probes to sort of understand the biology, but also to begin to really actually put together clinical trial plans that are meant to actually cure people. And some of the earliest attempts in combination therapy were discussed today. And so as we move from the lab into the clinic, we need to be thinking down the line as to exactly, what are we doing? Why are we doing this? Who are we doing this for? And how are we going to make sure that what we're doing now and in the near future is relevant to people living with HIV? And so that basically is going to require a deep, a long-lasting discussion between all the stakeholders, but particularly those people who are affected by HIV and who are the people who are trying to actually help. And so right now, what we're going to do over the next 40 minutes or so, we'll discuss some of the challenges of engaging a diverse community, why it's important. Hopefully we'll learn some tricks as to how best to do it. It's not something that we in San Francisco have been particularly good at, but we're trying. And we'll talk about this and some of the other challenges that we've confronted in terms of trying to build up a diverse participant population, and also to reach out to the community to educate that and make sure that there's buy-in, and we're all basically moving in the same direction. So we have a diverse group of panelists here. We have 5 people. They're each going to introduce themselves briefly, make some comments, and then I can basically follow up with some questions, but I really want actually people after these first 5 to come to the microphone with their thoughts, their perspectives, and their ideas, and any updates that might be happening at this meeting. So why don't we just start at the end? Tarira, why don't you introduce yourself and share your thoughts on that broad topic?
Thank you, Steve. I was hoping to actually be the last speaker, but I'll go ahead. I think one of the questions you asked us to think about is, how do we diversify cure studies and what are we missing? So I just had a few comments. This is always wonderful and engaging, and I think cure science is so important, not only for the field of HIV, but advancing our understanding of immunology, viral immunology, vaccine development, antibody development, etc. But I think it's really critical as we do this work to really make sure that we diversify the populations involved. So immune-based therapies, unlike small molecule ARVs that are really focused on the virus and can have broad implications across multiple clades, with immune therapies, the immune system is key. And making sure that we diversify that representation, so whether it's HLA diversity, whether it's different inflammatory environments, is really important. And for us working in HIV, Africa is one of the biggest regions affected by HIV. And as Africans, we're genetically diverse, with genetically diverse HLA alleles, exposure to various pathogens. So I think if our cure research is really focused on small populations in a small part of the world, we're really, as humans, missing out on the potential that can be learned from much more broad and diverse populations, and developing immune therapies that do work across broader populations. So I do think it's really important that we do more intentional basic science, basic immunology cure research in much more genetically diverse populations, as we see in African populations. The second point I'd like to make is that a lot of our cure studies are in somewhat ideal patients. And unfortunately, in the real world, ideal patients don't exist. So in our world, in the developing world, depending on where you are, up to 20 to 40% of people on ART started ART with advanced HIV disease, or CD4 counts below 200. It's very difficult and very rare to find acutely infected, acutely treated individuals without a lot of effort and a lot of intentionality. So our cure strategies have to be able to work in people with advanced HIV disease or people who initiated therapy with much more advanced disease. So really important as we think about diversifying trials. Yes, we want to have ideal populations, but I think we shouldn't think of it as an either or, but as an and. So yes, let's look at the ideal populations, get some wins, understand the biology, but at the same time start to think about much more diverse populations with more diverse reservoirs who started on treatment in later stage disease. The last couple of points I want to make is I think it's also important in the diversification to add adolescents. So in Africa, we have a large number of kids who are perinatally infected. Some of them made it through to adolescence and started ART when ARVs became available. So it's a fairly sizable population. And I think if we ignore that population, one, there's a lot of opportunities to learn about immunobiology in that population. But secondly, there are a large chunk of our society that is maturing into parenthood, and I think are the folks that we should be paying some specific attention to. And then lastly, I think the challenge here with what we're doing is that the research has to be iterative. Whether it's vaccine trials, it's highly unlikely that your first attempt is going to be successful, but really important to learn from it and adapt and move on to the next stage. And the only way to do that is by strong collaborative efforts. It's hard for pharma to do that iterative type of work. And I think it's really important as we design these studies to think about parts of the world that can also enable that rapid iterative learning. And unfortunately, the burden of disease is so high in our part of the world that I do think that there is opportunity to do some really important science that does allow for that rapid iteration and moving on. It's much harder to enroll a study with 1,000 HIV positive patients in Boston or San Francisco, but much more likely to do that in settings with high HIV prevalence where the need is also pretty significant. So I think the diversification can also enable us to better understand the challenges and dynamics for HIV cure.
I have a lot to add to that, but that was inspiring. But we'll let Jessica, has been working in this field and struggling with these issues for many years. Go ahead, introduce yourself and give us your perspective.
Okay, so I'm Jessica Salzwedel. I work at AVAC and I lead our HIV cure research efforts. And one of the things that I've been thinking about with this topic on how to diversify is really thinking about how do we create an enabling environment for engagement. I think, what all of you have sort of realized, right, is that the science doesn't just happen in a vacuum. There's policy and infrastructure that also needs to take place. And what engagement can really do is create an enabling environment of champions that can help push and accelerate the research. And creating that diversity is incredibly important because it brings new expertise and new areas into that. And, something that struck me throughout the meeting is, just hearing this idea of a cure for some or a cure for one that really is going to need to happen in order to lead to a cure for all. And so what diversifying engagement can really look like is helping both for communities to understand what that means and that it's this iterative process that goes in steps to get to a cure for all, but it can also help bring in that expertise that can really lead that and help enable that environment by being people who are experts in policy, who are experts in translating the research to clinicians and breaking down that science for all to understand. And something that we've done, and I guess I can go into this a bit more detail, is really helping with the academies to create that initial infrastructure that goes through a pipeline of first raising awareness and then building champions. So can talk about that a bit more after.
Sure. Okay, great. Okay. Krista, you have lots of stories to tell. Why don't you tell one or a few?
Hi, Hi. So we only have 2 minutes, so I'm going to start with my conclusions.
All right.
My conclusions are engage the community early. And by early, I mean during or even before protocol or study design. And my second conclusion is do it right. And do it right means engage the people that know how to do it, and that's social scientists. And some of us who are clinician scientists or just scientists, at worst, don't think social scientists are scientists. And so if you have that view, you're unlikely to really value their contribution or appreciate that they know how to do this and we don't. So engage people who know how to engage the community and how to bring the best out of them to our trials. So I work in KwaZulu-Natal in South Africa, and Dumbi and Dungu and I run the FRESH cohort, which is a cohort of adolescent girls and young women who we enroll before HIV infection, and we've enrolled over 4,000 young women now, and we've identified 101 hyperacute infections, and we're in the middle of running a CURE, or post-treatment control trial, using dual BNABs, VRC07 and CAP256, and a TLR7 agonist, vesitolimod. And I can say, when we were thinking about this, and we didn't start the cohort thinking about that we were going to have a platform for a CURE trial. We thought about it, well, we're going to contribute and learn about those early immunological steps that occur right after acquiring HIV. 5 years into starting the cohort, we thought about, wow, maybe we can actually run a trial here in the cohort. And it was at that time, years before we got the funding, that we engaged a social scientist. It was Corinne Dubé, who's dedicated her career toward thinking about HIV cure. So she flew to South Africa, and helped us engage the community, and those are community leaders, through a community advisory board and advocates, and started from scratch. What do you think about the word cure? What do you think about, what do you know about post-treatment control? And developing first a language that we could communicate back and forth with, finding out what they believed and what they thought. And FRESH didn't grow out of the ground as it is. We learned from the Thai cohort and their ECHO study. That's how we learned to even monitor and identify hyperacute infection. And when we went there first, before starting FRESH, it was amazing for the ECHO study. I've never seen any community that was so well engaged and was so supportive of participants. So that kind of stuck in our minds as we were starting things. But again, back to how did we engage at the time of the trials? We had discussed every little thing about what they believed, and they informed us about as gatekeepers for the community, who are real protectors of the young women that we were enrolling in the study, what things we should consider. And it was actually really eye-opening. And they were able to, before getting local ethical approval, before IRB approval, to look at the protocol, to look at the ICF, to look at the creating an ATI risk mitigation packet. So all of those things are going to make safer, more equitable trials. So again, engage them early and do it right, and allow us to be led, us as scientists to be led by those who know how to do it right, and let the community's voice and participant voices come forward and inform our trials, so they're real partnership and it's really patient-centered.
Melania?
I am going to speak from the perspective of the older woman who has seen pre-treatment, who has gone through AIDS illness, and who has experienced the Lazarus effect after treatment, and who participated in one of the first trials in Africa. And I speak wanting to represent women, but I do realize that I am from the older generation of people living with HIV, and therefore I will look at, my opinion will be guided more from my own lived experience. So, to start off, the subject of cure is an emotional one for anyone who has lived with HIV, who lives with HIV. I think I'm correct to say everybody longs for a cure. And that's the emotional side of it. But there's a practical side of it. And so I looked at it from what it was like for me when I agreed to go into a trial. I joined the DART trial, Development of Antiretroviral Therapy in Africa. We had two. We had Harare and we had Uganda. And we were looking at cohorts of 1,000 people, just trying to see whether art would work in Africa without the full resources of lab and everything that's required. What was it like at that time, and what led me to agree? And what is it like today? Why do I experience a hesitancy when it comes to cure trials today? Because that's what it is. I do have an excitement, but a hesitancy. And so I want to be able to manage these conflicting emotions. Back in 2003, when I joined DART, I didn't have much to consider. I had death in front of me. And I remember my doctor saying, well, you don't believe much in art, but you're dying anyway, so does it really matter? And so I didn't have much to consider. And I went, and time was not an issue because I had stopped work, I couldn't work. And I was a dependent, and therefore, really I had no plan for life. I was just there. And so I could spend days at DART while they did what they did, which I didn't understand much, but grew to understand as a layperson later. And today, a lot of what was discussed, I still don't understand much. A lot went over my head. But I'm held in awe that the whole world has gathered to address a cure for HIV. That is exciting, much as I don't understand it fully. But now today, the same science that has given me confidence to take art, to go through trials and take art, and the result is I have life and a good life expectancy at that, has created a certain satisfaction for the status quo. And so I would like to say, for the people that I represent, the long-term survivors, it is important for us to have confidence in science, in what it's doing. And I would like science to walk us through what is happening. A lot has happened today, but I would really long for it to be reduced to what is understandable for me as a layperson so that I can go. Similar to what the young ladies in South Africa might have experienced. So that I can be able to say, oh yes, there's hope there, there are setbacks there, but we are working it there. Let's go for it. We are all in a war. And I like to say I work at the HIV front, and I do have a passion for it, but I still experience this hesitancy to go into a practical and to join a trial. And then I think about women. I also represent more than half of all people living with HIV are women. And I was listening to all this and saying to myself, I wonder what this looks like for women. We talk about whatever, BNABs and whatever. I wonder what it is, what it's like for women. We talk about that cohort, and I say, I wonder what it is. So let's acknowledge that there are gaps. And let's get those gaps, let's address those gaps in treatment so that we are able to come to speed along with everybody else so that we can, with confidence, say, let's join science. They are for us. I'm getting emotional. I'll leave it at that.
So, Anna, you unfortunately have to follow up on that one.
Being the last
Yes.
is easy, but it's not, right?
No, it's not.
Okay, so let me present myself. My name is Ana Gabriela Pires. I'm a physician with residency in infectious disease and pediatrics from São Paulo, Brazil.
There you go.
I'm 10, right? but I have practice and directly care for people living with HIV in Brazil for many years. And 5 years ago, I joined AbbVie Incorporation in US and moved from tropical Brazil to cold Chicago. And since 2021, I have been the medical director for the AbbVie HIV program, which is in early phase. I think I'm here looking at you and your experience. I think I'm here more as a learner than really anything else. But I would like to share a bit our experience. I mean, our program is in phase 1. And we are looking for ART-free immune-mediated viral control. And for that, we have 2 programs. One, it's one of the potential candidates we are studying. It's ABV1A1, which is an anti-PD-1. And the other one is an alpha-4 beta-7 antibody, ABV3A2. And we did the phase 1 study, still is not complete. We don't have data yet, but it is fully enrolled. And I think we had several of the challenges that everybody has been discussing, right? And we learn from that, and I hope we're gonna learn even more to do it right? But some of the challenges that I would like to share with you. First one is anti-PD-1s are drugs that are being used to treat cancer. And of course, there are some adverse events that are specific to checkpoint inhibitors that we call immune-related adverse events that are very common in the oncology area. Oncologists are used to deal with, right? But not infectious disease. So I think one of the points that we learned during the study, and our study started in 2020, so in the middle of the COVID pandemic, which was not helpful. But one of the things that we learned was that the education is very important, right? Work together with the physicians, involve an oncologist if needed, involve people that have experience managing the immune-related adverse events was key for the study. And we really worked together, and it was exactly when we started to go more virtual because of COVID. So we really had to innovate this contact and really educate. The other point that we learned, as we learn with the good and with the bad things, is the enrollment was much more difficult and challenging than we thought. One point was the COVID, but other point was the fact, and you mentioned that, right? That I think we underestimate the need of communication and education of the potential participants, right? And really explain what we are researching. What's your chance of rebound? If you rebound, what's going to happen, right? And what you should expect. And I think this communication, it's very important to also, like you mentioned, right, to also help the participants on this emotional journey, right? Because you can imagine, of course, I'm just a physician, but I imagine that you join the trial and then let's suppose you rebound, you get scared probably, right? Because you are not expecting that. And how you handle that. So I think this is also another point which is very important, and we need to get better on that. So I'm really hoping I can learn to improve. How to really communicate in a way that the risks are very clear, how to mitigate those risks are very clear as well, and the expectations of what's going to happen, it's as clear as we can, because of course in a phase 1 you cannot anticipate everything. So this is my comments, and I'm open for any questions you may have. And thank you very much.
Thank you.
For the opportunity.
Okay, so we have a diverse group of panelists with different experiences and thoughts. I'm really hoping to have people in the audience who want to step up to the microphone and ask any questions or share their thoughts. Julie, you got there first.
Thanks so much, Steve. And I just really want to thank the panelists. I really think this was a fantastic discussion. And a number of really important points would be made. I just have maybe a couple of comments and then a question. One is just, I always take the opportunity to note with the diversification needed, to include sub-Saharan Africa, that we can't forget West Africa. Countries like Nigeria have a huge burden, and just because it's not as high incidence doesn't mean that those large number of cases, aren't critically a public health problem that need cure. And so that's something that at MHRP we've not been good enough doing cure work in West Africa, and something that we need to do more of. And I just think it's important for us to all remember that there's a big burden there, not just East and Southern Africa, which are of course still more gaps to be filled. And then I think, one other thing that was so interesting to me about the FRESH cohort was the economic empowerment aspect of that study. And so, the women who are enrolled, they actually, get skills that help them, it's a real contribution to their lives, their ability to probably makes them lower risk over time, right? But that is just really inspiring. And I think that to the extent where when we do these trials, if there's a way that we can be giving back to the community and to the participants, we're lucky because we have a PEPFAR program, so we can do that in the clinic, but not everyone can do that, right? So when you design those things that are part of it, I feel that just gives you a real ethical foundation for the work that you're doing and for the risk you ask people to do. And I just wanted to ask, Krista, if there's anything about that aspect of the FRESH cohort, that you've learned from in terms of good and bad points or challenges for incorporating those types of activities into that cohort.
Yep, FRESH is privileged and lucky, like you are with PEPFAR funding, to have a platform that we are able to establish long-term relationships with the participants. And so we're not recruiting from cold. There are participants in our trial that have been suppressed and with us for 8 years. So the requirement for the protocol is you're 12 months with suppressive ART, but we have 8 years of suppression and started in acute infection. So this is one of those ideal populations. But the thing about having an ideal population for your trial in our setting is they don't have ideal lives. And so what we've learned is they have extremely complicated lives. And if you're in one of these intensive post-treatment control trials, you can't work and you can't go to school. You really can't further your life. Everything's on pause for the duration of the protocol. And to appreciate that the observation period after getting the first dose of IP is 24 hours. And after BNAB infusion, it's 8 hours. And after dose escalation, it's another 8 hours. So these are incredibly intensive, and with an option of having lymph node biopsy, GALT biopsy, there's an incredible expectation on the participants, and then requirements of disclosure in a setting that has epidemic interpartner violence. So there's all kinds of considerations, and I guess the opportunity from having a program like the empowerment program is we have time to get to know the community and the participants better.
Add to that before you want to
Sure,
Steve?
yeah.
Just to that, I think that it's really important to say that investing in relationships with the community doesn't just have to be in an ideal cohort. There are ways to create that bond that allows for more people to not just become aware of the research, but also invested in the research. And again, I think that it's really important to think beyond just East and Southern Africa, but just to give an example from KwaZulu-Natal, CAPRISA has done a tremendous job of really investing in the community, building schools, investing in infrastructure that goes beyond just the clinical trial setting, and has a real investment from both the women in the community, but also their partners and their families and support structures that allow them to participate in prevention. And I think that those are some of the lessons that you can apply from prevention into the cure space more broadly.
Tori?
If I could also just add to that, I think it's also important, and we saw this with COVID and vaccine trials, et cetera, building trust with communities is really important. And the main point I want to make around that is absolutely we need to diversify. It's not just South Africa, Southern Africa, West Africa, et cetera, but also diversify the people who are doing the science, who are doing the research, have more local researchers, not just the research nurses look like people in the community, but the scientists, the people leading it, the people communicating about what we're doing at a national level, et cetera. So I think that's really important in building that trust, building those community relationships for this to work and to work effectively throughout Africa.
Okay, so we have 5 minutes, and we could probably go a little past that, so. Sharon, you're going to pass the baton?
It's a very quick question.
Yeah.
First of all, thank you. 5 absolutely fabulous stories. Clinician engagement's really important too. Jill Lau highlighted this in her presentation, but maybe Anna, speak to you about that with your trial. You were saying the challenge of the trial, but getting clinicians, how you work with the clinicians.
So I think one thing that worked super well is the peer-to-peer engagement, right? So it's not only involving other specialties to help the clinicians to diagnose and to handle, but it's also the experience. So we start to put people to discuss how I talk to my patient and explain for a potential participant about the trial, what are the information, how I follow up. And I think this was very helpful because it helped other people with less experience, other clinicians with less experience, to really have another type of conversation.
Yeah, no, I mean, I often find the clinicians are the most difficult people to deal with because they're fiercely protective of their patients, right? Which is a good thing. So, but anyway, Suzu?
Steve, and thank you, ladies. Really brilliant panel that you have, and I'm happy to see all the ladies on the stage. I think 2 reactions really to Melania's passionate perspective in terms of her journey with HIV and looking forward towards a cure. And one of the gaps that identified, I think, has been addressed by Tariro's latest response in terms of who's leading the science, especially for the African continent or other LMICs, who's asking the questions, who's driving the research, is the research coming from elsewhere, and we're just implementers, so having the clinical team or the nurses, and then having us as the community engagement, the face of the science. So if we can have more Africans or individuals in LMICs being resourced and supported to actually lead the research questions and lead the science, so that there's our communities can see that really this is part of us and it's not foreign and is from us. And some of the work that we've done with HCAP, which is the HIV Cure Africa Acceleration Partnership, is having a symposium tomorrow morning at 7:30. We will have breakfast, so please do come through. And I want to thank Sharon, Toombi, and Kathy, who will be part of our speakers tomorrow. We're looking at what is setting the target for cure, as well as leadership on the African continent. What are the insights that we've learned over the last 4 years of having HCAP and the partnerships that we've built with the sickle cell disease community, as well as the HIV cure community, including yourself, Steve. So part of, I think, Tariro's response that she articulated about African leadership is going to be talked about tomorrow, as well as the community engagement. Thank you. Oh, and we'll be in Auditorium Plaza at 7:30. Thank you.
And there could be coffee, I assume? No, everyone should be there tomorrow morning, 7:30. We will continue the discussion. Very inspiring way to start off the meeting. Thank you. Jeff.
Yeah, thank you to the panelists and to the organizers, especially for putting this together. I think it's very telling that as this got started, about a third of the people got up and left. And I think that speaks to the challenge we have in this field. And maybe the next time we do this, we should kind of sandwich it in the middle so that they don't have that.
I knew that was going to come up, yes.
Yeah, and thank you, Melania, especially for your amazing words. And as a long-term survivor myself, it's really frustrating to see that a lot of the trials have a very limiting exclusion criteria based on age, and it's an arbitrary number. And I think we really need to challenge the field to look at making sure there's a scientific rationale for each and every exclusion criteria, and not just saying you can't be 50 or 60 or 65, because the reality is, people over 50 are already the majority of people living with HIV. We're not going to have a cure tomorrow. It's going to be 10 years or longer. And by that time, everybody's going to be over 50, over 60. And we need to make sure that the science is done on the people who will be accessing that cure. It's absolutely vital. Thank you.
Did you want to comment? I mean, in your presentation, what came out to me in terms of the long-term survivor is the wisdom and the altruism, right? I mean, your lived experience has led you to sort of volunteer and think about volunteering things because of what you experienced when you were much younger.
Thank you. I also just was thinking, as you were speaking, that there is another challenge that is around the messaging that comes to people, and it's difficult to undo. Think about the childbirth, breastfeeding. There was a time when nobody could breastfeed, and it's hard today to be able to say it's possible to breastfeed. And so it's not easy to agree to a treatment interruption, because the message is take your tablet daily, if possible, timely. And so how do you undo that message? How do you undo it? And so these are the things that involvement becomes critical, that we begin to be able to say, is this what you're thinking as scientists? And this is why it's so hard for us.
Can I comment?
Real quick.
And I think this is fundamental, right? Because I think us as physicians and scientists, we are a little bit paternalist, right? So we try to exclude everything, so we are safe that, so how we really empower and educate people living with HIV to have very good informed decisions, and then we can be a little bit more, inclusive. I think this is the...
I would just say there's a difference between paternalism and infantilization.
Thank you. My name is Sina Temane. I come from Botswana. I'm an HIV/AIDS advocate, HIV Cure advocate. I trained in 2019 in Botswana, in Gaborone. I am a social scientist, and last year I implemented a program where I engaged the community, targeting religious leaders, traditional doctors, and the chiefs. Now, I want to speak to the aspect of resources, that resources are very, very limited. We have been trained to translate the tools to our vernacular languages, and even to the language that people speak. Like my mother was saying that sometimes packaging is a challenge, but we have been, as graduates from the academy, we have been trained. Now I want to speak to the stakeholders that have got the funding, they've got fed pockets. And I want to look at the manufacturers for ARVs, that where are they coming in? I want to see them giving back to the community, and I'm putting Jessica on the spot on this one, that yes, we need everybody towards the cure. We need funders, and I'm looking at the manufacturers for HIV. You are trying, you are giving us something, and we are treating HIV cure as cross-cutting issues in our organizations. That's really, pretty good. And I'm just saying, how are they involved? How are manufacturers involved in terms of funding, particularly? Thank you.
Jessica?
Yeah, I think that the funding for engagement is critical, right? And just like science, you do need to build an infrastructure that allows you the ability to build trust with communities. That doesn't just happen with one trial, it happens over time. And it's something that does need to be funded, but groups are doing it. And, I see, groups in the room like Gilead, which has a global community advisory board for Cure and is thinking about that up front, not just in their clinical trials, but for their whole program. And I think as Cure begins to evolve and expand, again, the lessons that we can take from prevention are that communities need to be involved at the basic science level and think about the whole program and investing in that from start to finish. But is there always room for more? Yes. And I think to say that, it's not fighting prevention or treatment. It's not, we can continue to expand that pie. And that's even more reason why we need to diversify the engagement and get more people involved. And, I think here we're thinking about the geographic region and talking a lot about sub-Saharan Africa, but I do just want to say that expanding the diversity doesn't just mean in Africa. It also means to the U.S. South. It means to women. It means to not just say that white MSM, which are predominantly those who have enrolled in trials and that we saw on the studies today, are not just the ones in the trials and on the community advisory boards, but are participating at all levels. And a lot of that right now is because people aren't aware, but when we can build that movement of engagement, that allows us to champion a single voice to pharmaceutical companies and others about needing to make more investments. But, just to say that those are there, and in that, plus in partnerships with the Martin Delaney Collaboratories, I think we are, and in IAS's panel with industry partners, I think that we'll see more investments in the future. But it's a really great point, Sina.
So, thank you. We're 7 minutes over. I'm getting a dirty look. Should we end this now? Yes, we should end. I apologize, guys. We'd love to hear your perspectives, but we're over time and we need to get going. And perhaps as many of us as possible can come to the HCAP meeting to continue this discussion. Thank you.
Hi everyone, we've got the next session now on considerations of language use for HIV Cure research, presented by Michael Luella. Michael Luella is proud to serve as a co-chair for the Delaney AIDS Research Enterprise Community Advisory Board. He has worked in HIV treatment research for 23 years, engaging communities affected by HIV in Seattle and across the US, in clinical trials towards a treatment and potential elimination of HIV from the body. Welcome, Michael.
Thank you, Heather. So I promise this will be quick, so just hold on. First of all, I just want to say we all kind of know that language matters. The words that we use have power, and we need to make sure that you put people first. And that is not just a philosophical sort of way we should be approaching clinical research, but it's actually a grammar rule that you should follow. Always putting people before the disease. People living with HIV, people living with diabetes, not saying HIV infected people or subjects especially. So think of that. People first, always people centered. This goes back since the beginning. In fact, 40 years ago, the Denver Principles were established, where you'll hear the language being brought up right away. It started off saying this: we condemn attempts to label us as victims, a term which implies defeat and which we are only occasionally patients, a term which implies passivity, helplessness, and dependence upon the care of others. We are people with AIDS. And you can hear those 11 men were actually saying something very powerful that has changed the way we practice research, the way we practice medicine, healthcare, all of those things, by putting the people first, people with HIV. So I've been a part of the Martin Delaney Collaboratories since they started, and we got community involved with that. The first three groups, Dare, Care, and Defeat HIV, were dealing with language from the start. The cabs of those three groups came together to come up with a cure glossary, and we're dealing with trying to communicate all of this complicated science to the communities that we serve. And we learned a lot of things about the terms that we were using, and I wanted to introduce one of them to you now. We didn't hear it much this time, thankfully, but sterilizing cure, which seems to be a problematic term, and we were trying to warn people about it. So it seems like researchers love this term, and you'll hear about it a lot in the literature, as they say. You'll find out about sterilizing cures all over there, but they seem to ignore the other part of the literature, which is really that there's other types of sterilization that are involved with HIV that are very negative. The forced sterilization of women, which is in the literature, but even if you want to look into the gray literature or the media, you can see sterilization has a negative connotation, especially if you're a woman living with HIV. This headline says it all from South Africa. We were trying to warn you about using sterilizing cure way back when. We were saying that this should be revisited as a term that's being used, but it didn't seem like a lot of people were looking at that sort of literature. We also had other things that were coming up. The idea of using the term functional cure, it seems to be an oxymoron. It seems to almost be saying almost a cure, not really a cure, but almost like it. And it's really communicating these things through the language that we're using that makes these choices questionable, especially if you're a community person, or especially if you're a person who you're wanting to maybe get involved in cure research, and then you hear these things, and they really don't seem to communicate. As we just heard earlier in the panel, we were talking about over 50% of the people living with HIV are women. Imagine hearing sterilizing cure. Sterilizing cure, does that mean this cure that you are creating is going to sterilize me? Is it going to make me infertile? Is it going to make me not able to have children? That's a big concern when you use terms like that. So we've been trying to come up with documentation for people that will help guide language choices, and this is a copy of something that we don't actually have here right now. We were going to have a printer give these copies so you would have them to take with you today, but we do not have that. So it will be available at the IAS booth in the Exhibition Centre once we get the copies made, so please look for a copy of this. It goes through lots of stakeholders' eyes and their viewpoints, and by stakeholders, not only just a global community of people, but we're talking about researchers as well as funders and other people who gave their input into what we thought would be considerations for the language choices that we have around cure and what we should perhaps use. So take a look at that. If you want to take a picture of this to get ahead, QR code is out there. Thank you for reminding me, Jessica. There's a QR code. So this has been looked at, and this is also a document that is in process. It's not a completed document, and so there's still time to give input and to really be a part of that. And I think actually, don't we have a webinar scheduled for September sometime where we're going to be talking about these things? So you'll be able to join us with that conversation that'll be going on in September. So these are just some strategies, especially for community, to help change terminology, making sure that your CABs know all about this, raising these issues at all meetings, not just at CAB meetings, but any sort of government or drug company meeting, keeping each other informed of these things. Make sure you go to the microphone if you hear something offensive, and also that we should be conducting studies or writing opinion pieces and to publish in the journals that talk about community acceptable terminology. Another great idea is something that the IAS did for this meeting, and I wanted to highlight it. They asked us to delete this slide, but I wanted to actually include it because I thought it was a brilliant way of bringing up language issues for people. So this was a first slide in the deck that they handed out to us, and they asked us to delete it before we actually talked about it. But you can see here, they went through do's and don'ts in terms of your language choices, and I think this is a brilliant idea. And if you're ever running a meeting, whether it's CROI or the Persistence Conference or just a small sort of regional or local conference, it's a great idea to actually give some guidance in terms of the language choices that might be affecting people when they're listening to what you're saying. So I wanted to thank the IAS organizers for this wonderful example that I hope is repeated from now until we have actually defeated HIV. And just to give a last little summation here, I think we need to let researchers know when they're using problematic language, and we can do that politely. We should have more patient-centered metaphors that focus on health and well-being, and we should also try to draw attention and understand and respect all the diverse linguistic, cultural, and community contexts that shape meanings and understandings around cure. And we also need more collaborations with community and social sciences to help us define what would be acceptable cure terminology. And with that, I am finished, and we have time for some questions, and if not, we can all get to the drinks a lot quicker. Thank you. Shall we go? Uh-oh. Oh, we have a question over there. Go ahead.
Good evening. My name is Sakabo Sisela Marumo. I am a young person who's openly living with HIV from Botswana. I'm also an HIV activist, advocate, as well as a research scientist. So my question was initially for Mam Tarira to share her lived experience, but since you're talking about language, I'll be asking, my question is in terms of going back to the community and sharing your findings as a researcher, what you found, that there are cohorts that were initially started or recruited some 10, 20 years ago, and papers keep being published even now, but the cohort participants are not being updated or told about the research. Then my question then becomes that shouldn't we make it mandatory that whatever paper is published, those people should go back and tell the cohort participants of what is happening and the findings in such a way that they can easily understand and not just read passively or be explained with big words that they cannot understand as people living with HIV. Thank you.
Yes. Yes, we should be doing that. Dissemination is a vital part of research. I think it's the part that we let go of the quickest. Maybe it's because we get too busy, but I think that's vital. If you want to have any sort of impact, bringing back your findings to the people that helped you get those findings, it just makes sense to me. And that's how you build trust. That's how you build engagement. That's how you build all the things that we were talking about earlier.
Hi. I just want to share my sentiments. Although my comment is originally for the last team, I feel like it can also benefit this one. I just want to share my experience as being a young researcher myself. There is a lot of stigma around HIV and AIDS research, especially in school. I remember when I did my first dissertation as an undergraduate, I wanted to do a research in HIV, but my professor changed it, saying that it has been overdone, and that you have to constantly defend yourself why you want to do HIV research. It's like you're disclosing your HIV status. But obviously, because I wanted to know more about HIV, going on to university, I remember when I did my master's, I was the only one who did HIV research in my group. And doing my second master's, I was the only one who also did HIV research. So when you look at the academic side of there is little HIV research that is being done in university, and for you to even do that research, you have to defend yourself constantly to say, why do I want to do this? Because they say it's overdone. And now, speaking of cure research and even the terminology that you're using, I don't think, speaking from my experience, there is no, in my university, there was nothing that related to HIV cure research. It's almost non-existent in universities. And coming here, this is my first time being at a scientific conference and being a postgrad myself. Most of the stuff that's being said here, we didn't know it even exists. And how are we then to continue this research pattern if we are to still have scientific researchers after the generation of older people has passed on? Young people are not in research in universities themselves. I know there's a lot of funding that goes around, and there are so many people that are paying for other degrees that probably won't even earn them into any job or something. And governments are now constantly looking at cutting programs that do not contribute anything in society, but you still find young people want to go to school. How are we then going to push for this, for young people to be interested in doing cure research? How are we pushing young people to be interested in just knowing about HIV beyond like HIV advocacy in the community? Because if there are no young scientists, if there are no young doctors, then I don't think we're doing much of a great job. So we can speak about this, but how are we then pushing this interest into young people, like going forward, like to just spark the energy of being like young scientists when there's constant stigma in the academic field?
good question. I don't have any answers for you, unfortunately, but it is something that's important. You shouldn't be silenced like that, and it's important to push back and to realize that there are generations to come that need us to continue this struggle that way. So that's really, I don't know if that's helpful in terms of my response to that, but I agree with you 100% that it is something that is necessary. And I think with that, we are out of time, and we should...
I'd just like to make one final comment, and it's about communicating the research to the community. And I'd just really like to thank Gillian Lau from as part of the HIV Cure Consortium in Melbourne on the Community Advisory Board for that group. And Gillian has instigated when researchers present their findings, having a community summary. And I was really proud to see that those summaries today. Fantastic. Thank you, Michael. And I hope all the other researchers take note, because there's so many that didn't do that community summary, and it would have been fantastic to have it to explain what all of that science meant. Thank you.
Thank you, Michael and Heather, for an absolutely wonderful session. There is no alcohol yet. We have one last part of the program, and hopefully this will help many of you in the audience, and that's our rapporteur session. And we have two fantastic rapporteurs. We've got Jill Lau from the Doherty Institute, who you've heard already a lot about. Jill's a clinician scientist, just finished her PhD, currently doing her postdoc with me on clinical research in HIV cure, and a strong interest in social research as well. And Ellen Bowden-Reid, who I believe is a PhD student at the Kirby Institute. And Ellen and Jill have shared the job of being rapporteurs, which is a tough ask when they're coming up straight after the end of the meeting. So we'll hear from them, then we'll have some closing remarks from one of our co-organisers, Beatrice Moat, and then there'll be drinks and posters. Over to you, Jill and Ellen.
Thank you, Sharon, for the introduction. So we won't introduce ourselves. But... No. Yes, so this has been a very hectic but very interesting day. So it started with Sharon, the president of IAS and co-chair of the Towards a HIV Cure initiative, for setting the scene and the goals for the day, which should be on a slide. But essentially they were to try and establish a unified definition for post-intervention control, understand the biological impacts of immunotherapies for cure, understand the underlying mechanisms, as well as learn from the advances in immunotherapies in the field of cancer and also in vaccine development. And then we had a very nice welcome from Heather Ellis from Positive Women Victoria to welcome the community, and shared her story about living with HIV for 27 years and also being diagnosed prior to ART, and also Google. So. Then we had heard from Ranjini Thomas, who gave a keynote from an autoimmunity perspective, and I think this is important to allow us to think more broadly to the solutions that we're challenging in both HIV research and autoimmunity. And it really highlighted a lot of the parallels between finding an HIV cure and treatment of type 1 diabetes and also rheumatoid arthritis. And then in the context of antigen-specific tolerizing immunotherapy, which Ranjini has really pioneered for rheumatoid arthritis, there was a lot of key considerations, again, which are similar between the fields, including having that antigen, that efficient antigen presentation, as well as TCR recognition, and also reversing exhaustion, and that has certainly been spoken about in abundance today. Go closer so I can read this. Sessions 1 and 2 focused on clinical trials. So we're talking about both of them together. Clinical trials and animal studies exploring immunotherapies such as broadly neutralizing antibodies, HTI vaccines, which are HIV-specific T-cell immune response vaccines, with or without toll-like receptor, JAK inhibitors and immune checkpoint inhibitors. And it was really exciting to see a pipeline of clinical trials being presented with early data from the Rio long-acting BNAB study, a collaboration between Rockefeller, Imperial, and Oxford universities, presented by Ming Li, and the UCSF multi-intervention study, lovingly known as JAWS,
presented by Michael Peluso. In each of these studies, a participant was able to suppress virus for a prolonged period of time, and these investigators also acknowledged the challenges in conducting complex and intensive trial protocols, and this included invasive procedures such as gut biopsies and repeated leukapheresis after many years of treatment interruption, which was presented as well in that session. We are seeing more BNABs, not just in the HIV cure space, but also in treatment and prevention, HIV trials. And a recurring theme and discussion this morning was the ability of BNABs to control virus beyond the expected decay or half-life of these agents, the washout period of BNABs. Is this a vaccinal effect that was discussed a bit this morning, or of these agents, or do they need to be more deeply understood? In contrast, a non-human primate study presented in session 1 by Binghua Ling did not show a delay in viral rebound beyond the expected decay of the BNABs in her study. Other important discussion points this morning around BNABs were the discrepancies with sensitivity testing and the need to develop more broadly applicable BNABs to the global population of people living with HIV. A pertinent question was raised around whether we should be targeting subpopulations more likely to achieve a cure, such as those with very small reservoirs or those with specific BNAB susceptibilities, or to generate this signal. But this small signal may not be replicable or applicable to broader global populations. So, what is the right balance? And I think this is something that definitely needs to be explored and discussed going forward. Most of the clinical studies presented this morning reported eventual viral rebound in the majority of trial participants when treatment was paused in an analytical treatment interruption. However, this wasn't the only endpoint of these clinical trials. The research presented throughout the morning described sub-analyses in both human and non-human primates studies. Work presented included immunological assays to identify biomarkers of immune activation and expansion that predicted viral decay from Monica Rees, pharmacokinetic and pharmacodynamic studies of BNABs shared by Michael Peluso, transcriptomic signatures of HIV-specific CD8 T cells to predict responses, and reinvigoration of HIV-specific immune responses reported by Jan Schroeder. And Beatrice Mothe showed us that we can predict HIV viral control in HTI vaccine studies in participants with high magnitudes of HIV-specific T cell immune responses. Finally, another important point raised is that we are that we ensure that all these studies are using appropriate sensitivity assays to measure effects such as viral rebound and latency reservoir characteristics, as emphasized by Tokame Mahmoudi. Sorry, we have no slides, guys. Oh.
Should we wait? Just keep going. Right. I'll just keep pushing on so everyone can get their drinks. Oh, we're there. How do you go to the next slide? Oh, thank you. That one? Oh, yeah. All right, so we'll just quickly skip through. This was the boring but interesting one, as Ming Lee presented. Where are we up to? Here we are. All right, so session 3 was the gene and cell therapy session. We really saw some interesting strategies that have been presented in this session that are currently being used to address a lot of the challenges that we're facing with HIV cure research. And these, again, have certainly been discussed here at length today. So Paula Saval presented a modified novel targeted lipid nanoparticle formulation to address the challenges that we have with delivery and getting these therapeutics to really difficult cell types, such as resting CD4 T cells, and also proposed a next generation LRA that could potentially be used to overcome the potency challenges that have been observed with previous LRAs. And then David reiterated, the challenges that LRAs have had and continue to have, and how potently reactivating the reservoir such that we're able to see measurable differences and reservoir clearance has really been an ongoing challenge. Eileen Boritz presented data on the Chicago patient, and I think this is also, interesting, as we've just seen the Geneva patient come out in the news in the last couple of days. And this was the first documented case of viral rebound after a Delta 32 mutation stem cell transplant. And this was first presented at CROI this year as a poster where we were given a more detailed understanding of the case. And so this person had a stem cell transplant for AML in 2021. And then single genome sequencing identified that there was no X4 virus present pre-transplant, which was very interesting. And then the patient underwent treatment interruption 15 months after their transplant, and viral rebound was observed from week 8 onwards. But again, only CCR5 tropic virus was identified using their assays. And so 6 people have now been cured to date. This is the issue when you're reading off notes, which includes the Geneva patient. And as mentioned, this was the first case to demonstrate viral rebound. And even though all other factors were similar to previously described cures. And then there were no HIV-specific immune responses as well measured to pre-treatment interruption, but this case demonstrates that this does not necessarily predict success in this case, unfortunately. Cool. And so then the final session, which is probably still quite fresh in your minds, was on post-treatment control, which was the 4th session of the day. And with reported rates of post-treatment control or post-intervention control ranging from 10 to 30%, as we heard in some of the earlier sessions of the day, there's really a great push to understand the mechanisms that are underlying this, which is obviously the theme of the session. And so while we still don't completely understand why some people are able to suppress HIV or low to undetectable levels when they're no longer on ART, but presenters of this session really tried to describe, they did describe novel methods to try and kind of unpack this all. And so this included some single NK cell analyses that we saw by Keith Reeves, and the PEARLS assay that was described by Sarah Palmer, which found genetically distinct viral populations and mutations in predicted T-cell epitopes, which might be a way to predict post-treatment control. And then immunophenotyping by Tongsu Ma identified CD8 positive TEMRA cells, which were increased in post-treatment controllers across all treatment interruptions. And then, as expected, they also had lower frequencies of exhausted CD8 T cells.
There were some really interesting non-human primate studies presented today. So firstly, there was really promising work demonstrated by Ziwei Chen from China of a non-human primate model with anti-PD-1 vaccine. So I'm really excited to see the future work of that in clinical studies to come. Timing matters, so neonatal non-human primate model essentially was reported to show a functional cure with a single dose with a bivalent antibody. So once again, really interested to see what this progress into the clinical world. And Sophie, acknowledging the challenges of implementing broadly neutralizing antibodies due to their half-life, proposed B-cell editing for prolonged expression and demonstrated that B-cells can be efficiently engineered to express anti-HIV broadly neutralizing antibodies. Finally, Shelby O'Connor described an amazing ability of Mauritian macaques to exhibit post-treatment control. So let's watch this space and try and understand what her group is able to, what these monkeys can do, and see if we can understand that from a human perspective. We will just briefly touch on the last two sessions. We were furiously typing away and trying to put our rapporteur session together, but it was an amazing panel of all women talking about the challenges, but also just how do we address and do better when it comes to clinical trial engagement and improving diversity of participants in cure trials, not just gender equity, but also making sure older people are included, adolescents are included, and also how scientists embrace and harness the lived experiences of people living with HIV to improve HIV cure design and conduct. We have a picture of Michael, but not much to add. This was 5 minutes ago, so Michael is always a force to be reckoned with, and his real call to arms to really mind our language and communicate effectively and efficiently and with care when it comes to reporting HIV cure science, but also the design and how we word clinical trials. And I think I'm really appreciative of IAS pushing that with the language, a people-first language stance they've taken for this conference. I'm still going? Yep. Themes that we've noted today. A few recurrent themes. There's an emphasis on the role of combination interventions. So I think Michael Peluso made a comment that a single intervention by itself is probably not going to cure HIV, and you've seen just a number of clinical trials of combination therapy presented today. Another theme that we noticed was not noticed, but really hammered home was the importance of engaging community throughout the lifetime of a clinical trial. We noted challenges of conducting this research during a global pandemic and the delays that resulted from this, but how these were overcome by various investigator teams presented today. And interventions were discussed, but there's also an importance of the cohorts, people behind these studies, so not just the drug or strategy. Does the cohort matter? So, for example, people started very early in HIV infections, such as phoebic stage 1 or 2, maybe that's my main example, highlighting not just biomedical strategies to cure HIV, but the importance of the individual patient factors. And finally, samples from even failed studies. We've seen a number of examples of old samples being used to really try and understand mechanisms of persistence or control. And I think it's important in this world to not waste samples that are collected, particularly difficult to obtain samples such as tissue biopsies. So it's really heartening to see that work continue to be used.
And then finally, we'll just wrap it up again with the forum goals that Sharon described at the beginning of the day. And, I think, we think some of these have been achieved and some probably still require a bit of work to do. Thank you for your attention. Apologies for reading the notes, but absorbing this in a day would be impossible. Thank you, and we look forward to seeing you at the remainder of IAS.
Okay, so no slide, don't worry. So I have to only thank everyone for standing and staying until now. I'm Beatriz Mote. I'm one of the co-chairs of the forum. It's been a great experience for me being part of this organization committee. I would like to thank that. It's been exciting for all this last month. I would like to thank all the participants of the forum that have traveled from both near and far to the forum and that will stay for the IAS meeting. Thanks to all the program committee that have been working, as mentioned before, during all this last month to put this program, which I think it's been very challenging to put everything that we wanted in one day on what's going on cure research, and thanks all the IAS staff, again, from helping in setting up the forum. And thank all the speakers the panelists and also the rapporteurs. I think it was a very difficult task to summarize everything that happened today. And I think that it's more, I won't summarize again what it has been told. So the last thing is that I was told that I should share with you that there's the launch of a CFAR AfriCure program that basically it's ambition to support a new generation of scientists from Africa. I think this is one of the important things that were discussed during the last panel. We cannot do research here and then go to Africa to do the trials, but science needs to be done there, and we need programs that can boost those scientists from Africa to be engaged from the beginning and to the end of all the cure research that we are developing. So I think that's great news that this program is being launched. I think in September there will be open some grantees for scientists to get some funding to do this. And well, lastly, I would finally invite everyone to join the poster exhibition. I think is the moment to network everyone with some wine and some food. So I hope you enjoy the last part of the meeting. Thanks, everyone.